Imagine a world teeming with life. Much of it completely unseen, yet it's profoundly impacting our crops, our health, even the very air we breathe.
It really is. And today, we're diving into that hidden world.
Yeah, specifically focusing on a truly fascinating and honestly often surprising group within the fungal kingdom.
It's a realm that's critical to understanding everything from really devastating plant diseases to some surprising medical conditions. But, it often goes overlooked.
Absolutely. So get ready for an in-depth look at the Ascomycetes. These are, well, a diverse and incredibly important group within the phylum Ascomycota.
And our mission today really is to unpack their unique structures, try to demystify how they reproduce, and discover their sometimes quite dramatic real-world impacts.
For this deep dive, we've really dug into a fantastic chapter from an Introductory Mycology textbook.
Yeah, it's great source material. Think of us as your guides, sort of sifting through the dense details to pull out the most intriguing nuggets for you.
By the end of our chat, we hope you'll have a new appreciation for these often overlooked organisms. I mean, from the microscopic mechanics of spore release
To that billion-dollar lesson in agricultural history.
exactly, and even the fungal culprits behind certain human infections. There's quite a lot to unpack.
So where do we start? How do these hidden organisms actually operate? What's their, unique fingerprint, the thing that sets them apart?
Okay, so the defining characteristic, what truly sets Ascomycetes apart, it really revolves around their asci, the specialized sacs where spores are produced.
Right, the spore sacs.
Yeah, and the key here is that these asci develop within preformed cavities, almost like tiny, custom-built rooms inside a larger fungal structure called an ascostroma.
And what's really fascinating, I think, is that this ascostroma, this sort of protective housing, it's already there before the asci even begin to develop.
Right, like the house is built and then the furniture, the asci are moved in.
Exactly. And that's a fundamental difference from how some other fungi form their spore-producing structures. Often the walls develop around the asci, not before them.
Yeah.
So it points to a very specific developmental pathway.
So it's all about that preformed cavity.
Okay.
And beyond that preformed home, they have another crucial feature you mentioned, the batunicate ascus. That sounds a bit technical. What does batunicate actually mean in practice?
Well, batunicate literally just means two-walled. So this ascus has two distinct layers, two walls. They're separable.
Okay, 2 layers.
And this unique double-layered structure is absolutely vital to how the spores get released. The outer wall, it kind of ruptures, splits open, and then the inner wall actually expands forcefully like a piston right through that opening to shoot the spores out.
Whoa, so it's almost like a spring-loaded mechanism, like a tiny, perfectly engineered launch pad for the spores.
You've got the image perfectly. Yeah, it's often described as a jack-in-the-box mechanism.
Yeah.
The inner wall layer pushes out, extends, acting as a dispersal tube, and it launches the spores out into the environment. It's remarkably efficient.
That's such a vivid picture. A fungal jack-in-the-box. And these ascostromata, the homes for the asci, they can be pretty varied too, right? Not all the same.
Oh, absolutely. They can be incredibly diverse in form. Some are multilocular, meaning they have many of these cavities, these locules, within that larger structure.
Okay.
While others are unilocular, containing just a single cavity, and when they're unilocular, they're often called a pseudothecium.
Pseudothecium. Why pseudo?
Well, pseudo meaning false, because it can look very similar to another common fungal structure, a parathecium, just looking at it superficially. It's tricky even for mycologists. You really need to study its development, how it formed, to know for sure it's a pseudothecium, formed within that pre-existing stroma.
Right, you need the backstory, the developmental blueprint. Okay, so within these tiny fungal homes, the arrangement of the spores and any supporting tissues can also vary a lot. Mycologists call these centrum types.
That's right, centrum types. Think of it like different architectural blueprints or maybe interior designs for their spore-producing structures.
And why is this internal architecture so important, just for classification?
Well, yes, for classification, but also because these centrum types define the precise arrangement of the asci and any other sterile tissues within that locule. They reveal deep developmental patterns and evolutionary relationships. For example, some designs, like the Alcinoe type, are quite simple. You just have these globose, sort of round asci scattered in the locules with no other sterile tissues around them.
Okay, pretty basic.
Yeah. And the spores get out after the asci themselves burst through the stromal tissue covering the locule.
And what about others? Are there more complex internal setups?
Oh, definitely. Other types, like the Pleospora type, are more intricate. Here, the asci are intermingled with something called pseudoparaphyses.
Pseudoparaphyses. Again, with the pseudo.
Right. These are sterile hyphae, kind of like tiny downward growing support threads or filaments. They actually originate above the spore-producing area and grow down among the asces.
Interesting. Like internal scaffolding.
Sort of, yeah. And these precise internal arrangements, down to the microscopic level, they're incredibly important clues for how we classify these fungi and understand their lineage, how they're related.
What's fascinating is that even the Botunicate ascos itself, that two-walled structure, isn't always uniform. Are there like subtle variations even within that design?
Yes, absolutely. There's quite a bit of microscopic variation. Sometimes it's about how exactly the two walls separate. The fistotunicate type is the classic jack-in-the-box where the thin outer wall clearly ruptures. But there are other types like semi-fistotunicate or rostrate where the separation isn't quite as complete or happens differently.
Hmm.
And you can even find tiny modifications, the very pip, the apex of the ascus, things called an apical chamber or an ocular chamber.
These sound like really minute details.
They might seem trivial, yeah, but for mycologists, they're like unique signatures. These subtle differences are surprisingly important for understanding their evolutionary relationships. They help scientists piece together the family tree of these fungi and figure out how different groups carved out their own ecological niches.
It's amazing to think about these microscopic structures having such importance, but all that intricate design, it serves a purpose, right? Reproduction and survival.
Exactly.
So how do these fungi actually go about multiplying and spreading? For many loculo-ascomycetes, it seems asexual reproduction is maybe more common than sexual reproduction, especially for plant pathogens. Why would they lean on that?
You're right. For many, particularly those causing plant diseases, the asexual form, what we call the anamorph, is often the dominant phase you'd encounter in the field.
The anamorph.
The sexual form, the teleomorph, might only appear much later, maybe after the host plant tissue has died, or sometimes it remains completely unknown for certain species.
So why favor the asexual route?
Well, asexual reproduction is just a very fast and efficient strategy. It allows for rapid multiplication and widespread dispersion using spores called conidia. That's a huge advantage for a pathogen trying to spread quickly during a plant's growing season. Maximize infection when conditions are right.
Makes sense. Quick and easy copies. But when they do reproduce sexually, how does that typically happen? I read about something called spermatization. What's that about?
Spermatization. It's a
Yeah.
fascinating process. In some species, like Mycosphaerella tulipiferae, you have these tiny, non-motile, male-like cells called spermatia.
Spermatia.
And they can actually fuse with specialized receptive female hyphae called trichogynes, which often extend out from the developing fungal structure.
So it's like pollen finding an ovule, but fungal style.
Kind of. This fusion acts as the trigger, the fertilization event for the sexual cycle.
Okay.
It initiates the development that eventually leads to the formation of the ascocarp, the structure housing the asci, and the production of the sexual spores, the ascospores. It's a clever way for them to exchange genetic material and maintain diversity.
Okay, to really bring this life cycle stuff to life, let's talk about a classic example. Apple stab. The fungus Venturia inequalis. It sounds like it perfectly illustrates this complex fungal strategy. What happens there?
Right. Apple scab is a great example. This fungus, Venturia inequalis, is heterothallic. That just means it needs two compatible mating types, kind of like sexes, for sexual reproduction to occur.
Okay, needs a partner.
Exactly. So imagine springtime. Apple buds are bursting open. Now, hidden in the dead apple leaves that overwintered on the orchard floor.
From last year's infection.
Precisely. Inside those dead leaves, the fungus has completed its sexual cycle over the winter. And now, ascospores are forcibly ejected from the pseudothecia up into the air.
Launched by that Botunicate ascus.
You got it. Those ascospores are carried by wind, maybe rain splash, to the newly emerging susceptible apple leaves or blossoms. If there's enough moisture, they germinate and infect.
Okay, so that's how the infection cycle starts each spring, from those overwintered leaves.
Exactly. Once it infects the leaf, the fungus grows under the cuticle, and pretty quickly, within days sometimes, it starts producing huge numbers of asexual spores, called conidia.
The anamorph stage.
Right, the anamorph stage, which for this fungus is called Spilochia pomi. And this stage is incredibly efficient for spreading the disease. These conidia are washed or splashed by rain onto other leaves, or young developing fruits causing secondary infections. This leads to that rapid spread of scab lesions you see throughout the spring and summer.
A real snowball effect. And then the sexual part of the cycle kicks in later. How does that happen?
Yes, typically late in the growing season, maybe as the host apple leaves start to die off in late summer or fall, the fungus then penetrates deeper into the leaf tissues.
Ah, so it waits until the leaf is on its way out.
Often, yes. And then it slowly begins to form the ascostromata, the pseudothecia we talked about. Inside these structures, the sexual process occurs over the fall and winter months. Compatible mating types fuse, undergo genetic recombination, and eventually produce the new generation of ascospores that will be ready for release the following spring.
Wow. It's a beautifully adapted cycle, isn't it? Perfectly timed with the apple tree's own life cycle and the changing seasons.
It really is. It highlights how fungi adapt their life cycles, very specifically to their hosts and their environment.
It's amazing to consider how these tiny organisms have such intricate strategies for survival.
Yeah.
But okay, what does all this intricate design and these life cycles mean for our world? Do these fungi have a significant impact on, say, agriculture or even our health?
Oh, they absolutely do. A huge impact. Many Ascomycetes are notorious plant pathogens that can cause and have caused widespread economic devastation.
Like the apple scab we just talked about.
Yes, exactly. Venturi inequalis is a globally significant pest, costs apple growers millions every year.
Ah.
But there are others too. Gignardia bidwelli, for example, causes great black rot.
Great black rot.
Yeah, this fungus is actually native to North America, but it was accidentally exported to Europe, probably on infected grapevines in the 19th century, and it had absolutely disastrous effects on European vineyards, which had no natural resistance.
Wow. Another example of introduced species causing havoc.
Precisely. And then you have Mycosphorella species. These cause various leaf spots on bananas and plantains, like black sigatoka disease. These are economically destructive, seriously impacting a major global food source, especially in tropical regions.
It's sobering. But the billion-dollar lesson of corn blight, that really brings home the economic impact, doesn't it? Tell us about that because it sounds like a really profound story about genetics and agriculture.
It really is a powerful and, yeah, very expensive lesson. So back in 1970, the United States experienced a catastrophic epidemic of southern corn leaf blight. It devastated the corn crop, particularly across the U.S. Corn Belt.
What caused it?
The culprit was Cochleobolus heterostrophus. Specifically, its asexual form, the anamorph, called Bipolaris mates, but a particular strain known as race T.
Okay, raised tea. What was special about it? Was it just unusually aggressive?
It was more than that. Its devastating impact was a direct consequence of a widespread agricultural practice at the time. Farmers were relying very heavily on a single genetic trait in hybrid corn.
A single trait in most of the corn.
Yeah, it was called Texas male sterile cytoplasm, or CMST. Using this trait made producing hybrid seed much easier and cheaper because it eliminated the need for mechanical detasseling, removing the pollen-producing tassels from the seed parent plants.
Ah, so it was a convenience thing for seed producers.
Exactly. And by 1970, something like 80%, maybe even more, of the U.S. corn crop carried these specific CMST mitochondria. The problem was, race T of Bipolaris mates produced a very specific toxin called T-toxin.
And this toxin targeted.
It only attacked the mitochondria of plants with that CMST cytoplasm. It didn't significantly harm corn with normal cytoplasm. In the CMST plants, the toxin caused the mitochondria to swell, leak essential molecules, and basically shut down respiration.
Oh, wow. So the very trait that made seed production easier made the plants incredibly vulnerable to this one specific fungal strain.
Precisely. The result was massive crop losses, estimated at the time to be over $1 billion, which was a huge amount then. It was a powerful, incredibly expensive reminder of the dangers of genetic uniformity in agriculture. Relying too heavily on a single genetic pathway creates enormous vulnerability.
Putting all your eggs in one genetic basket.
Exactly. It really underscored the critical importance of maintaining genetic diversity in our food supply to buffer against unforeseen threats like new pathogen strains.
That's a truly profound lesson learned the hard way. But okay, these fungi aren't just about causing disease, are they? You mentioned they also produce fascinating secondary metabolites. What does that mean?
That's right. Like many other fungi, Ascomycetes produce a whole range of complex chemicals that aren't essential for their basic growth, but serve other functions: defense, competition, communication. We call these secondary metabolites.
And some are harmful.
Some definitely are. Certain Alternaria species, for instance, produce mycotoxins, fungal toxins that can contaminate cereal grains like wheat or sorghum, causing discoloration or making them unsafe for consumption by humans or livestock.
Right. But others can be beneficial.
Yes. Some produce compounds with potentially useful properties. For example, presummerins, which are isolated from Priosia, a fungus often found on dung, have shown promising antibacterial and antifungal activity in lab tests. Research is ongoing into these kinds of natural products.
Interesting. And then there's some really weird effects, like sportsmen. You mentioned this earlier. Are these fungi literally making sheep sunburned? That sounds wild.
It sounds bizarre, doesn't it? But yes, sporidesmin is a toxin produced by Leptospirulina chartarum, which often grows on dead pasture grasses. When grazing animals, particularly sheep and cattle, ingest forage contaminated with this fungus, the sporidesmin damages their liver.
Okay, liver damage.
And one consequence of that liver damage is that a breakdown product of chlorophyll, normally processed by the liver, builds up in the animal's bloodstream. This compound makes the animal's skin extremely sensitive to sunlight, a condition called facial eczema, or more generally, photosensitization.
So they get severe sunburn on exposed skin.
Exactly.
Hmm.
Especially on the less pigmented areas like the face and ears. It can be a major animal health problem in places like New Zealand and Australia during certain times of the year. It's a remarkable, if unfortunate, example of the potent biochemical power these fungi wield.
Wow. Okay, so beyond pathogens and toxins, many Ascomycetes are also endophytes. What does that mean for their role in the environment? Are they like hidden collaborators inside plants?
Precisely. Endophytes are fungi. They live inside plant tissues, leaves, stems, roots, but without causing any obvious signs of disease. It's often a mutualistic or commensal relationship.
So they just hang out inside the plant.
Pretty much, yeah. And this is a hugely active area of research right now. Scientists are discovering an increasing number of these endophytic loculoascomycetes, especially as they explore biodiversity in more regions, particularly tropical forests.
Like finding new species.
Exactly. For instance, Letidropsis palmorum was only relatively recently described as a common endophyte living inside the leaves of Amazonian palm trees. We're constantly finding new ones.
What's the significance of that?
Well, it highlights the vast undiscovered biodiversity out there. And these endophytes might be playing really important ecological roles. Maybe they help protect the plant from herbivores or other pathogens, or help it tolerate stress like drought. We're only just beginning to understand these complex hidden relationships. It's a reminder of how much more there is to learn about the microbial ecosystems all around us and inside other organisms.
That's fascinating. Hidden helpers. Now, while they're often known for their plant interactions, you mentioned that some Loculoscomycetes are significant animal and even human pathogens. Can you give us some examples of their impact closer to home on us?
Certainly. It's important to remember they're not just plant specialists. For instance, species of Piedrea, specifically Piedra horte, caused a condition called black piedra.
Black Piedra.
Yeah, it's basically hard, dark nodules that form, encrusting the hair shafts of primates, including humans. It makes the hair feel gritty and can be quite irritating, though it's usually just a superficial issue.
Okay, so more annoying than dangerous. What about more serious infections?
Well, then you have members of the family Herpatriculaceae. This group includes fungi, often referred to collectively as black yeasts because of their dark pigmentation and often yeast-like growth in culture. These can be opportunistic human pathogens.
Black yeast, that does sound a bit ominous. What kind of infections do they cause?
They typically cause infections after some kind of skin trauma, like a puncture wound. This can lead to conditions like chromoblastomycosis, which results in chronic, thickened, warty skin lesions, sometimes with subcutaneous cysts. It's often difficult to treat.
And can they get deeper?
Yes, unfortunately. More severe systemic infections affecting internal organs like the brain and lungs can be caused by species such as Cladosporium trichoides, now often called Cladophila forabantiana, and Exophiala dermatitis. These are particularly dangerous for individuals with compromised immune systems.
That's serious. What's interesting is where these black yeasts actually live naturally. It's not exactly common places, right?
No, it's really fascinating. Their natural habitats are often quite extreme, specialized low-competition environments. Think intermittent brackish water, slimy drain pipes, crevices in rocks, even places with high salt concentration or temperature fluctuations.
So they're tough little survivors.
Exactly. It suggests they're highly adapted to surviving in harsh conditions where other microbes might struggle, which, in a way, makes their ability to sometimes switch gears and cause infections in humans, another challenging environment, even more intriguing from a biological perspective.
And it's not just these specialized black yeasts, right? Didn't you say some of those plant pathogens we talked about earlier can also cause problems for humans?
That's a really critical point, yes. Some of those fungi, primarily known as plant pathogens, belonging to orders like the Pleiosporellus genera, such as Bipolaris, Alternaria, Curvularia, can indeed cross over and cause infections in humans.
What kind of infections?
It can range from relatively superficial things like skin lesions or nail infections, onychomycosis, to more invasive conditions like sinusitis, keratitis, eye infections. And in immunocompromised patients, they can cause even more serious, life-threatening conditions like brain lesions, encephalitis, and severe lung infections.
Wow. So the lines can be blurry between plant and human pathogens sometimes.
They really can be. Medical mycologists actually sometimes use specific fungicides, like Benomol, which inhibits certain fungal groups more than others, as a diagnostic tool in the lab to help differentiate between some of these morphologically similar fungi when they're isolated from a patient. It's a complex landscape connecting environmental fungi and human health.
Okay, so given all this incredible diversity, these complex life cycles, the varied ecological roles, and these surprising impacts, it kind of makes sense that figuring out how to classify Ascomycetes is, well, complicated and always changing. What are the challenges for scientists trying to sort them all out?
It is absolutely a dynamic field. Classification or taxonomy is a constant process of reevaluation. Scientists are continually re-examining relationships. Traditionally, this was based heavily on morphological characteristics, the shape of the ascostroma, the type of centrum development, the details of the
The
ascus.
things we discussed earlier.
Exactly. But now, increasingly, molecular evidence, especially DNA sequence data, is playing a huge role. And sometimes the molecular data tells a different story than the morphology alone.
So the family tree gets rearranged.
Frequently, this large group, the Ascomycetes, encompasses numerous different orders, like the Pleiosporales, Dothididales, Hysteriales, Astronales, each with its own unique variations. For instance, the Hysteriales have these elongated, slit-like ascostromata called Hysterothecia, while the Astronales have flattened, shield-shaped ones called Theriothecia.
So much variation, it sounds like maybe they aren't all as closely related as their shared features might suggest.
That's precisely the puzzle. Molecular studies are strongly suggesting that the entire group of fungi traditionally lumped together as Ascomycetes based on having bitunicate asci developing in locules within an ascostroma. Well, it may not actually be monophyletic,
Well, monophyletic meaning.
meaning they don't all share a single, unique, common ancestor to the exclusion of all other fungi. It implies that some of these key features, like the bitunicate ascus or the ascostroma, might have evolved independently multiple times or been lost in some lineages. It's called convergent evolution.
Ah, so similar solutions evolve separately. That makes classification much harder.
It does. It highlights that scientific understanding is always evolving. What we define as a group today might be redefined tomorrow as we get more data. It's an exciting, ongoing process of discovery in mycology, constantly challenging our assumptions about fungal relationships.
What a journey. We've really dug deep into the intricate world of Ascomycetes today. We've uncovered their unique structural adaptations, from that cool jack-in-the-box bitunicate ascus.
Yeah, and the diverse ascostromata, those preformed homes, and even the different internal architectural styles, the centrum types.
We've seen their cunning reproductive strategies too, whether it's that rapid asexual spread using conidia or the more complex sexual cycles involving things like spermatization, all perfectly timed to their environment and hosts.
Like that apple scab example, adapting perfectly to the seasons.
Exactly. And maybe most importantly, we've explored their immense real-world impact as devastating plant pathogens that literally taught us billion-dollar lessons about genetic diversity.
A hard lesson, that one.
Yeah. And as producers of both harmful toxins like mycotoxins or that photosensitizing sporidesmin, but also fascinating beneficial compounds.
And even as endophytes living hidden inside plants and as pathogens affecting animals and humans, from black piedra on hair to serious systemic infections caused by black yeasts or even crossover plant pathogens.
It's incredible. These microscopic organisms truly shape our world in profound and often really surprising ways.
They really are masters of adaptation, silently influencing so much around us. It's truly amazing how much biological complexity and power is packed into such tiny forms.
So maybe the next time you see a spot on a leaf, or even just, a patch of mold somewhere, consider the hidden complexity and the surprising power humming away within the fungal kingdom.
Yeah, think about what other unseen organisms are profoundly shaping our environment and maybe even our health in ways we're only just beginning to understand. There's always more to learn.
Absolutely. Always more to discover right under our noses or inside a plant stem.
That's right.
Well, thank you for joining us for this deep dive into the fascinating world of Ascomycetes. We really hope you gained a new appreciation for these often overlooked but incredibly important masters of adaptation.